Okay, so thank you for coming for my talk. I'll be telling you a little bit about the work that I have done during the past year on the evolution of social polymorphism in Formica group of ants. So as many of you may already know, ants are one of the most species-rich organisms on the planet, and they have been incredibly successful in colonizing numerous types of habitats. How do I minimize that? Yeah. And they've been able to do so mainly by division of labor, which is the characteristic feature of ant societies. And this is brought about by numerous castes of ants that are present inside a colony. Here I have two examples. Camponotus on the left, where you can see a lot of physiological differences between the castes, and Herpagnathus on the right, where the castes look quite similar. Now, among these castes, we have a queen, which is the reproducing individual in a colony. She lays eggs. The males are short-lived and usually are produced during the breeding season. They mate and they die soon after. We have the workers, which are also female, and they do most of the work inside a colony, from feeding and raising the brood, foraging for food, as well as protecting the colony. Soldiers are more specialized workers whose main job is to protect the colony from invasion. And on the right, we have queens and workers in the Herpagnathus colony. In this case, both of the castes look quite similar to each other. And in some species such as this, in the absence of a queen, the worker can switch on reproduction and start behaving like a queen and start laying eggs. So the life cycle, I'll briefly show you the life cycle inside a colony. So when queens and males mate, a mated queen can produce two types of offspring. Usually when they establish a new colony from scratch, they first produce workers and soldiers, which form the bulk of the colony. And when the colony has attained a certain size, they can start producing sexuals, which are the breeding individuals, which will mate and establish new colonies. So we have division of labor, a single reproducing queen, and this poses an interesting evolutionary question regarding cooperation. So we've learned in evolutionary biology that individuals that contribute more to the offspring in the next generation or contribute more to the gene pool in the next generation are selected or are successful. So in this case, how does social organization work, where workers and soldiers are not reproducing themselves, but helping another individual reproduce? So this could be explained by the theory of kin selection. So as you can see, all the individuals inside a colony are quite related to each other, are highly related to each other. And so even though workers and soldiers are not reproducing themselves, they're indirectly passing on their genes by helping the queen reproduce. And this delicate cost-benefit balance is what maintains social organization and cooperation. So there is an added layer of complexity to social organization in ants, because in some species, you can have colonies which have more than one queen. So you have single queen colonies, and you can have multiple queens inside the same colony. And now this adds another layer of complexity, because when you have multiple queens which are unrelated to each other, the relatedness inside a colony can drop dramatically. However, this also has its benefits, because with multiple reproducing queens, rapid colony growth can be achieved within a short period of time. Similarly, single queen colonies can be beneficial under different circumstances. So queens from single queen colonies can disperse better because the colony only has a single queen. Future queens will establish a new colony elsewhere in the habitat, so they are able to colonize new habitats. Whereas in case of multi-queen colonies, because more than one queen can be accommodated, they tend to form satellite extensions of the original colony rather than colonizing an entirely new habitat. So when you have abundant resources and a stable habitat, the multi-queen strategy can be quite beneficial. Whereas when the habitat is more unpredictable and resource availability could be scarce, their single queen colonies could be more successful. So this is a very interesting system to study, where you can actually look at which factors affect or help cooperation, and at what points does social organization start to break down. The species that I study, Formica selesi, has single queen and multi-queen colonies. I'll be referring to the single queen as monogynous or M colonies. and multi-queen colonies as polygynous of P in the remaining talk. So in Formica selaceae, the switch between monogynous and polygynous colonies is genetic, and a large portion of chromosome 3 in this species is responsible for defining the social form. And as I have color-coded the regions which are involved in this genetic architecture, and among the two haplotypes, as they are called, among the M and the P haplotype, they have undergone genetic rearrangement. So depending on which combination of haplotypes an individual inherits from the parents, they could either behave like the single queen or the multi-queen organization. So in Formica group in general, there are numerous species which exhibit this type of social polymorphism. And they all share the genetic architecture which is responsible for this. So it's thought that the genetic architecture evolved before these species started diverging from each other. And so it has evolved about 60 million years ago. And the two species that I'll be talking to you about are Formica cinerea and Formica selaceae, which are quite closely related to each other. So when I started in the lab, I had the whole genome sequences of these species. And so I started with looking at how these sequences are related to each other. So I constructed a phylogeny like you see here, but with the data that I have the species color-coded on the left. And what I observed when I looked at the genome-wide sequences was each species formed a separate cluster. And so, and Formica selaceae and Formica cinerea are closely related, so they form sister branches. When I looked at the same, but only the regions responsible for the social polymorphism, what I was expecting was to see something like this. Now, because the genetic architecture is older than the species themselves, I expected to have two groups for each of the architecture, and within that, species-specific branching. But what I saw in the data for the monogyne architecture, it was according to what I expected. But for the polygyne architecture, now the Formica cinerea, which is in orange, has split into two groups. One which clusters inside Formica selaceae, which is in yellow, and the second which is outside, which was the original expectation. So this led to two interesting observations and hypotheses, which is that Formica cinerea probably has two different types of P haplotypes, one which is similar to Formica selaceae and one which is slightly different. And that the similar P haplotype has probably come from Formica selaceae into Formica cinerea. So this is quite interesting for these two species because we know that they hybridize. So along the Rhone Valley in Switzerland, you will see there is an entire region where both these species co-occur, and you also have hybrid colonies which are a mosaic of both Formica cinerea and Formica selaceae. And why should we care about this? Why is it interesting? Is because this is in fire ants, which are a highly invasive species. It is shown that hybridization and introgression is when you have regions of genome which are exchanged between species through hybridization. In fire ants, hybridization and introgression has led to rapid spread of polygyny. So fire ants are invasive. They have rapidly spread to several parts of the world, and they are a threat to farmlands, crop yields, and newborn animals, which is why they are a pest. And there are extensive measures being taken to curb their population growth. And in case of fire ants, polygyny has spread quite recently, within the last million years, across several species through hybridization. And even though Formica are not invasive and pest species like fire ants, but it's interesting to compare these two phenomena because here the social organization and the genetic basis is quite old. It is about 60 million years old, whereas here it's fairly recent, in about the last million years. So it's an interesting system to study the population dynamics of social polymorphism. And it also provides an interesting scenario when our Formica cinerea has two different P haplotypes, which can lead to questions such as what maintains the haplotypes in the species, why doesn't polygyny just take over, why is monogyny still there, and so on. So what I did first was to look at whether introgression has occurred. And to do that, I performed a test using a D statistic. Now, what D statistic does is it is when you compare different regions of the genome between two groups, or in this case, two species, it looks at regions which show higher similarity than would have been expected from their relatedness. And I have done this comparison for both M and P haplotypes for Formica cinerea and Formica seleci. And what I've shown you two comparisons because I've looked at movement at introgression in both directions, from seleci to cinerea and cinerea to seleci. So in both cases, you see that the P haplotype has a higher similarity or is more shared between the two species compared to the M haplotype. And I also looked at the opposite. So I also looked at genetic divergence. So genetic divergence between haplotypes which is shared would be lower, and between haplotypes which are not shared between two species would be higher. So I looked at the genetic divergence between the two M haplotypes. Here I've used FST as a measure of genetic divergence. And this is chromosome 3. And I've shown you the region which has undergone rearrangement between the two haplotypes, which regulate social polymorphism. And so for M haplotypes, you see consistently high genetic divergence between the two species. But for P haplotypes, you see a drop in genetic divergence, which is right about the region which has been rearranged. And so this shows you that the P haplotype between these two species is more similar than would have been expected and has likely been introgressed. I would also like to, I will be performing these comparisons with the other P haplotype as well, which was in the Italian populations, which is distinct from the one which is shared. And I just have 2 samples for that so far. So I need to add more samples to this. And recently we've collected more samples which are of this kind, and we'll be sequencing them to add more data to this tree. So to summarize, I've shown you that the P haplotype, which is responsible for polygyny, has probably introgressed from Formica seleci into cinerea, and that introgression can help rapid spread of polygyny. And I will be trying to understand how these two haplotypes are maintained within the species. And with new sampling, I'll be hopefully able to get more data to answer that question. And with that, I'd like to thank you for your attention. This is my lab, and I would also like to thank all my lab members and former and present lab members who helped me with this data. Thank you.